We'll continue describing the cell movement during amphibian gastrulation. Gastrulation in frog embryos is initiated on the future dorsal side of the embryo, just below the equator in the region of the gray crescent. Here, the cell invaginates to form a slit-like opening, as you can see from the figure, which is called the blastopore. These cells change their shape dramatically. The main body of each cell is displaced toward the inside of the embryo, while cells maintain contact with the outside surface by way of cylindrical neck cells called bottle cells. And you can see these bottle cells in the figure. It is the very first orange cells that have been internalized. These bottle cells will line the archenteron as it forms. So you'll see that the orange cells will start to line the internal as they internalized. Thus, and the invagination of cell initiate the archenteron formation, and you can see from the figure that archenteron is developing while the blastocele is reduced. And you can see from the figure from the outside how the cell converge from the outside to the inside. The gastrulation in frog begins not only at most of the vegetal region, but in the marginal zone as well, which is not seen in the sagittal section, but you can see from the outside view how the cells from the marginal region is migrating. But from the sagittal section, you only see the cells migrating from the dorsal and the ventral hemisphere. When these migrating marginal cells reach the dorsal blastopolar rib, they run inward and traveling along the inner surface of the outer animal hemisphere, as you can see from the figure. Thus, the animal constituting the lip of the blastopore, either dorsally, ventrally, or marginal, are constantly changing because they push inward and another cells are replacing its place. The first cell to compose the dorsal blastoporal lip, as we said before, which is the bottle cells, they lead the other cells to internalize, and they represent the leading edge and form the leading edge of the archenteron. And these cells will form in future the pharyngeal cells of the foregut. As these cells pass into the interior, the dorsal blastoporal lip become composed of cells that involute into the embryo, forming the precaudal plate cells, precursor of head mesoderm. So that means these precaudal plate cells is replacing the involuted cells, which is the prospective pharyngeal cells of the foregut. The next involuting cells that replace the precaudal plate cells is the chorda mesodermal cells, which will form the notochord, which is a transient mesodermal backbone that play an important part in distinguishing the patterning of the nervous system. As you can see from the figure, you have the orange cells which migrate first, and followed by the pink cells, then followed by the red cells, and you can see that the bottle cells directs these cells for the internalization, and you can see that the blue layer of cells, which represent the presumptive ectoderm, trying to make an epipoly and try to covering the embryo from outside. If we elaborate more on the regions of the gastrula, you can see that the blue will form the prospective epiderms, while the orange will form the prospective mesoderm, and these type of cells are have the gene cerebrus is expressed, while dark orange will form the prospective head mesoderm and characterized by the expression of the gene GU2, while the red part, which will give the presumptive chorda mesoderm, characterized by zebra gene expression. At the beginning of gastrulation, the involuting marginal zone IMZ forms, which is composed of chorda mesoderm and the head mesoderm. The vegetal rotation, as you can see from the arrow on the figure, the vegetal rotation pushes the prospective pharyngeal endoderm to the side of the blastocele. You can see in C and D that the vegetal endoderm movement push the pharyngeal endoderm, which is represented by the orange color, forward, driving the mesoderm passively into the embryo and toward the animal pole. The ectoderm as well begins to start epipoly, pushing and trying to covering the embryo from the outside. So in this figure you can see that most of the endoderm have been internalized. The ectoderm, represented by the blue color, is almost covering the whole embryo, and the archenton is well developed, which will form in future the gut, and the blastocele is highly reduced. And as the new cells enter the embryo, the blastocele as well displaced to the side opposite to the dorsal blastopore. Meanwhile, the blastopore lip expand laterally and ventrally, and we can see this from the figure from the outside. Initially, it is the dorsal blastoporal lip, then start to migrate laterally to form a crescent shape that extend laterally and ventrally until merging to form a complete blastopore. And through this blastopore, you can see the endoderm, which now is termed a yolk plug. And one of the main characteristic or feature of completion or the end or termination of gastrula is the reduction of the size of the yolk plug. With the formation of the ventral lip, the blastopore has formed a ring around the large endodermal cells that remain exposed to the vegetal hemisphere. And you can see from the figure, it shows how the movement occur. So you can see that the arrows, when it's solid, it represents the movement from outside, while the arrows which is dashed represented the movement inward. So the remaining patch of endoderm now is called, which appears yellow in this figure, appears, we call it, it's called yolk plug, and will eventually be internalized. You can see here that gastrulation almost have been done in the first figure, and from the sagittal section and dorsal view of the embryo, you can see that another process is starting to occur, which is the neurulation. It starts with the formation of a neural plate and the neural fold, and it is completed by the formation of a neural tube. So once the gastrulation finishes, at that point all the endodermal precursors have been brought into inside of the embryo. The ectoderm has enriched the encircled the surface, and the mesoderm has been brought between them. And you can see here the developmental stages of the cleavage followed by gastrulation, looking to the embryo from the outside, how the cleavage occurs followed by the formation of blastopore, dorsal blastoporal lip, and the crescent form, and then the migration of the dorsal blastopore to form a yolk plug, and then starting of the neurulation and formation of a neural tube. In this figure, you can see the different developmental stages during neurulation: early neurula, mid-neurula, and late neurula from the dorsal side. And you can see how you can distinguish between these three stages by the stages of the neural plate. In the early neurula, neural plate is flat, and then the neural fold in mid-neurula start to migrate to the dorsal side until the complete fusing of neural fold to form the neural tube. In the mid-sagittal section, you can see how the neural tube is formed in the mid-neurula, being lined by two layers of the dark blue layer of cells, and in between is the neural tube. And in the late neurula, you can see that the neural tube is covered by the ectoderm, which is light blue. In this section, you can see that in this figure, you can see the transverse section and how the neural plate have migrated with its neural fold. The neural fold migrated dorsally and until complete closure of the neural tube and the complete covering of the neural tube by the ectoderm, the light blue. And you can see that as well that the mesoderm have been well developed between the ectoderm and the endoderm, and the starting of the cavity of the gut have been formed. In this slide, you can see the difference between autonomous and the conditional development in the gastrula. You can see in the first experiment, the tissue is transformed between two early gastrulae. They took the region of cells which will give the presumptive neural ectoderm, and they implanted these cells into a region which will give a presumptive epidermis. The embryo generated only one embryo with one neural plate, because the region that they have taken will initially it will presumptively give a neural ectoderm, but when they are transferred to another region, they decided to make an epidermis according to the location, while they repeated the same experiment, but in this case the only difference, they used a late gastrula, not early gastrula, and they do the same setup, but the embryo developed with two neural plates. That means that the cells that have been implanted have been developed into two neural plates and promoted another neural tube formation. So you can see here what is the difference between autonomous and the conditional development. In another set of experiments, they transferred a region that will, the dorsal blastoporal lip tissue from an early gastrula to another early gastrula and transplanted this in the region that will normally becomes a ventral epidermis. But in this case, they found that the region which have the transferred, the implanted tissue, they develop a second archentron, and the second embryonic axis is formed with a secondary neural tube. That means it took the memory of the location of it and induced a secondary structure. So the embryo that will arise eventually, a second embryo will arise with a jointed embryo, and you can see at figure D, it's a live twin Xenopus larva generated by transplanting the dorsal blastoporal lip into the ventral region of an early gastrula. In this slide, we'll see the amphibian mesodermal differentiation. You can see early after the formation, after the nucleation process, the mesoderm is generally divided into two regions, the somatic mesoderm and the lateral plate mesoderm. Soon after that, the lateral plate mesoderm will be differentiated into two regions, an outer somatic layer which is near the ectoderm and an inner splanchnic layer which is near the endoderm. There is a cavity in between them that is called the splanchocele. Soon after that, the somatic mesoderm will be differentiated into three main regions: the dermatome, which is near the ectoderm, outer dermatome, and an inner sclerotome, which is near the notochord, and in between the myotome, which will develop into muscles. There is a cavity in between between these three structures is called the myocele. We can see here how the mesoderm have been developed with a cavity inside the mesoderm, which is called initially the splanchocele, and then will give rise to the coelom, which will give rise to the peritoneal cavity, which will presumably surround the organ. Another structure appeared well defined, which is the neural crest, which is actually the neural fold have been, the tips of the neural fold have been separated and then got located between the neural tube and on the dorsal side between the neural tube and the ectoderm. And this neural crest will be important in the formation of a lot of structure later on. This slide summarizes the developmental stages of the amphibian development, starting from the fertilization until forming of the limb bud embryo, which is near hatching. So you can see how the cleavage proceed, followed by the mid-cleavage state, late cleavage state, then the formation of the dorsal blastoporal lip at stage 10, then the mid-gastrula, late gastrula, and the neural blade formation starting from stage 13, until the complete closure of the neural tube ended by stage 16, then formation of a tail bud, muscular responses, and then the heart beating starts, stage 19, and then the gill starts formed at the stage 20, and the operculum is formed at stage 24, and finally the limb bud stage 37.